Hello, welcome back. We are going over session 6, genetics. Session 5 was a little bit on the short side, and session 6, genetics, does cover a lot more material. So let's take a look. We are revisiting the differences between meiosis and mitosis. Remember, meiosis is the creation of those sex cells. They do have the potential to have that information passed on from parents to offspring, which is why we are talking about it in Session 6, genetics. We're also going to be taking a look at Mendelian and non-Mendelian patterns of inheritance. So remember the things we learned from Gregor Mendel, like some alleles are dominant, some are recessive. You inherit your characteristics from your parents. Those are all Mendelian genetics. And then the non-Mendelian things are like codominance, incomplete dominance, sex-linked traits, polygenic traits, and multiple alleles. Don't forget, we talk about how chromosome number can be altered through problems like non-disjunction, deletion, insertion, and inversion. We also go through the process of protein synthesis in session 6 here. It was unit 7 in class. But in protein synthesis, we're going through transcription and translation. So we're going back to that central dogma information about how DNA transfers its information onto messenger RNA and then proteins, and proteins code for a trait. So again, going through the central dogma, DNA to RNA to proteins, proteins code for a trait. Okay, we're kind of stuck here. I can't get out of that slide. Okay, then we're also going to talk about how genetic mutations can alter the phenotype. You have your silent mutation, your nonsense mutation, and your frame shift mutations. They may or may not impact the phenotype of an organism. We're going to explain how genetic engineering has impacted the fields of medicine, forensic science, and agriculture. And those are our main ideas. So there's a lot of information to cover in this unit. Some videos that I thought would be really valuable for you to take a look at are the Khan Academy video where he does review Punnett squares. It's a little bit lengthy, but you really need to make sure that you look at Punnett squares and you remember how to do them. Another reminder, make sure that you're using your scrap paper. When you take the Keystone exam, the scrap paper is there for a reason. You'll see other people are just like, I'm not going to use it. But even I use scrap paper when I think about these questions. It really helps you to be able to just kind of make a little drawing or write little reminders for yourself. So when you come across a question that involves a Punnett square, make sure you use your scrap paper. The other video that I have for you is Mr. Anderson going over Mendelian genetics. So that's a good one to watch. The next two videos are me going through the protein synthesis draw along. This is where we talk about the central dogma and we talk about the process of making proteins. And then over here, we have this guy on the internet and he talks about the actual transcription and translation and shows us some of the effects on the phenotype that different mutations may have. Your questions for you to answer for this unit were Mendel's 3 basic laws of inheritance, the 5 non-Mendelian patterns of inheritance, protein synthesis and the central dogma, which I recommend you do a drawing for, and how mutations may or may not affect the phenotype of an organism. Let's get into the practice questions. Practice question number 1. Which statement best describes how an organism uses DNA and amino acids to synthesize a protein? So what you need to remember for this question is that the word synthesize means to make a protein. A, DNA is transcribed into messenger RNA, which is translated by the ribosome to produce an amino acid chain. DNA is translated into messenger RNA, which is transcribed by the ribosome to produce an amino acid chain. Amino acids are transcribed into messenger RNA, which is translated in the nucleus to produce a molecule of DNA, or amino acids are translated into the messenger RNA, which is transcribed in the nucleus to produce a molecule of DNA. You know what I'm going to say. Vocabulary is so important. Make sure you have a good working knowledge of the vocabulary from Unit 6 and Unit 7 to be able to answer a question like this. Take a moment to answer this on your Ed puzzle. So for number 1, if you selected A, you were correct. The two processes of protein synthesis are transcription and translation. So how we make proteins by transcribing the DNA into messenger RNA and then translating that information at the ribosome to build amino acid chains called proteins. A is the only correct answer for this. Everything else just doesn't make sense. Number 2 is a question that does require you to spend a little bit of time thinking about how we transcribe messenger RNA into amino acids. I'm going to switch over to the pen for a second. I have to tell you some test taking strategies here. Remember that you have as much time as you need to take the Keystone exam. It is not a timed exam. You can go beyond the time allowed. We strongly recommend that you go through the test once and then go back and make any changes or corrections after you go through the whole exam. You're going to want to make sure that you're using your scrap paper and you're taking your time. Don't rush. Settle in, relax, and do your best. A question like this is going to take anyone a few minutes to answer. And some of these questions will take you a few minutes to answer. Now, we saw this in Unit 7 when we were transcribing the amino acids. What I don't like about this question is that they only give you this type of amino acid chart. They don't give you the round one. And some people just really prefer the round one, and they're both widely accepted. So we're going to practice with this one. So which mutation would most likely affect the organism's phenotype? So which one is going to be seen in the physical appearance of your organism? Each one of these bold letters is a mutation. So our first sequence of amino acids is AUG. AUG is going to code for methionine, MET, and that is the start codon. That's always the start codon, okay? So all of these are the same, no problem there. In letter A, we have G, C, C, okay? G, C, C. That is coding for alanine, A, L, A. In letter B, you have G, C, G. That is also coding for alanine. So ALA here as well. And can I write with my hand there? ALA. Okay. And then we've got G, C, U, which also codes for alanine, A, L, A. And then finally, the same thing, G, C, U. So alanine. So far, no differences, even though there's a mutation at the third codon in letter A and letter B. So far, they're both coding for the same amino acid, so we're not going to notice a difference. All right, so methionine for all of these, alanine for all of these. Okay, U, G, U is going to give us cysteine. UGU, same thing, cysteine. Okay. UGA gives us a stop. Okay, so that's going to be different. And then UGC gives us cysteine again. Okay, so for A, it's methionine, alanine, cysteine. B, methionine, alanine, cysteine. C, methionine, alanine, stop codon, which is going to shorten the amino acid chain. And then finally, methionine, alanine, cysteine. So C is going to be our correct answer here. It took a few minutes, and that's okay. Key that into your Ed puzzle. Okay, number 3. Let's look a little bit closer at this. I thought this was a little bit confusing because you really need to pay attention to where it says start of replication. Right here where it says start of replication. And when I first looked at this question, I actually was looking at it from the top down because that's how you like naturally read something. And what? No, that's not what I want. And so when we look at this, we need to read this from the bottom up, which again, why would they try to make it more confusing? So T pairs with A, C pairs with G, C pairs with G, A pairs with T. We always follow the rules for base pairing. So T pairs with A. Here we've got an A. C goes with G. C goes with G. A goes with T. So following the rules for base pairing, you should be able to conclude that A, G, G, T is the most correct answer. It's not A, G, G, U because uracil replaces thiamine only in ribonucleic acid. This is clearly DNA because it's double-stranded and there's T's in it. I'm going to try not to give you the answer on the next one. Which activity would a cell be unable to perform if a mutation occurred that caused the Golgi apparatus to stop functioning? If the Golgi apparatus stopped functioning, we talked in session 3 about the role of the Golgi apparatus. So you have to kind of think back to that. Would it stop packaging proteins for transport out of the cell? Stop processing messenger RNA as it is transcribed from DNA strands? Would it stop providing the location for the delivery of amino acids by transfer RNA? Or would it stop producing a template to guide the sequencing of amino acids? Take a moment to think about this question and answer it on the Ed puzzle. If you selected A, the packaging of proteins out of the cell, you would be correct because when I think about the Golgi apparatus, I think about the post office. So the Golgi apparatus is the post office for the cell, and that's going to go ahead and package everything up and get it ready for transport. I do need to jump back to I skipped one. Okay. I guess I'm giving you the answer to this one too. The genetic event shown in the diagram can occur during meiosis. Which statement best describes this type of event? We talked about this a lot of times. Okay, gene sequences that are identical are replicated. Sections of DNA between non-homologous chromosomes are exchanged, decreasing genetic diversity. Segments of homologous chromosomes are exchanged, resulting in cells that will be genetically different, or the traits from two sections of the same DNA strand are altered, resulting in the production of a new species. What? Whoa, that was a leap. That was a leap. If you see the letter C and think that seems like the most correct answer, you are correct. We know that crossing over, this occurs during meiosis 1 of occurs during metaphase 1 of meiosis, and this is going to increase genetic diversity, creating organisms that are genetically different, and that's why C is the most correct answer. All right, I got us out of order a little bit, and I do apologize. So we're going to jump down to number 7. In order to study genetic mutations, scientists must study genetic material. Which statement describes the genetic material scientists are most likely studying? So if we are studying genetic mutations, did I skip another one? It's okay, we'll go back. If we're studying genetic mutations, which type of material are we most likely studying? The alleles that contain chromosomes, which are RNA, alleles that contain genes, which are chromosomes that contain DNA segments, which are RNA, or chromosomes that contain genes, which are DNA segments. All right, so answer that in the Ed puzzle. Okay, what we have here, if you selected D as the answer, you are correct. Alleles that contain chromosomes that are RNA, that makes no sense. DNA is also not RNA. That makes no sense. Alleles contain genes, which are chromosomes. This is a little bit backwards, okay? So D is the most correct answer. Again, I've gotten this slideshow all out of order. I'm trying, guys. All right. Which statement describes how cloning is different from selective breeding? So getting into the genetic engineering part of this. Cloning eliminates recessive alleles from a genome. Cloning transforms harmful traits into beneficial traits. Cloning corrects or replaces mutated gene sequences, or cloning results in offspring genetically identical to the parents. All right, vocabulary term for you. Answer this question in the Ed puzzle. You know that a clone is a genetically identical offspring. So 74% of students in Pennsylvania got this answer correct. So you should be able to as well. All right, we did number 7 already. Let's go to number 8. Okay, another one where they just give you like a weird diagram that doesn't make any sense. Let's take a look at this together. DNA to RNA to proteins. We know that this is the central dogma. What does Y represent and what does Z represent? Is Y replication and Z is translation? Translation, replication, transcription, translation, or translation, transcription? Take a moment to answer this in the Ed puzzle. If you selected C, you are correct. DNA is transcribed into messenger RNA, which leaves the nucleus of the cell, gets translated into proteins at the ribosome. So Y is transcription and Z is translation. Number 9. Which statement best describes the process by which the millions of body cells that form a housefly can all contain the same genetic information? So it has nothing to do with houseflies. It's just saying, when you look inside of your cells, your skin cells and your bone cells and your muscle cells, they all contain genetic information that is unique to you. Let's think about why. Original DNA is duplicated during replication and then distributed into 2 new cells. Original RNA is duplicated during replication and distributed to 2 new cells. Original DNA is duplicated during replication and distributed into 4 new cells, or original RNA is duplicated during replication and then distributed into 4 new cells. This is a little bit going back to the differences between meiosis and mitosis in chapter 5. Think about this question for a few minutes and answer this in the Ed puzzle. If you selected A, that is the most correct answer. Think back to when we were building the DNA molecules. We had our original strand used as a template for the 2 new strands. This happens during the S phase of the cell cycle, and it produces 2 genetically identical daughter cells. So this happens from second number 1 of, All right, so DNA is duplicated during S phase of the cell cycle, DNA replication, and then distributed into 2 new genetically identical cells. RNA is not replicated, and then DNA doesn't make 4 new cells. That would be meiosis. So A is your most correct answer. And then finally, this was a long one. Again, scrap paper, babies, scrap paper. Use the diagram below to answer question number 4. The DNA section goes from A, G, T, G, C, C, G, A, C in the original strand. The altered strand is A, G, G, C, C, G, A, C. A section of DNA in a cell is altered. Which mutation is being illustrated in the DNA section above? Is it a deletion, insertion, a duplication, or non-disjunction? This is a tough one. Think about it for a minute. Use your scrap paper. Answer the question in the Ed puzzle. Okay. Okay, if you selected deletion, you are correct. You can see we have an A and a G. The T has been deleted. So now the G moved down, the C moved down, the C moved down, the G moved down, the A moved down, and the C moved down. The T has been deleted, shifting the rest of the sequence down by one nucleotide. Okay, insertion would have the opposite effect. Duplication and non-disjunction don't apply to this type of a mutation. All right, that'll do it for this session. That was a long one. That's a lot of thinking. There's a lot of material to cover here. If you're still confused about any of this, make sure that you watch the videos that have been provided for you, and also that you're going to the Biology Keystone exam webpage for our school, and you're taking a look at the practice sheets that are provided for you to review from. All the vocabulary is in there. There's cloning, right? We talked about that. All of the vocabulary is in here, and just some pictures and samples and other video recommendations. So a lot of good resources if you are still confused. Thank you.